Good. All right. Well, mostly. There we get down.
I don't care about that last little bit, I don't think.
That's gonna.
Okay,
Good.
good.
It's close. All right. Yeah, let's zoom in on the, I think, what is a Chrysogorgia on the left there.
Okay.
After you stow.
Thanks. Okay, you want the one just below the lasers now?
Yep.
Okay, go ahead, Ed.
There's like 3 or 4 of them around here. Wow. I haven't seen this one yet.
All right, happy there?
Yeah, let's see if the one, there's, I think there's a couple to the right of the sponge. Let's see if it's the same thing.
Yeah, they're a little further away, I think.
Yeah, I might have to
hop up to image them.
Yeah.
Whoa, I gotta go back to F lens cap.
F lens cap. Yeah, I think they're just behind the arm now, so I'll jump up and move.
Oh, here, I'll leave the sponge exposed properly.
Sure.
And then hop up and wag right and I won't touch it. You'll see where we were light-wise.
Yeah, there's a lot of them here.
These ones here?
Like midnight.
Or
right,
the
let me
3
fix
right in
that.
front of you? Or?
Yeah, can I do the one to the
There you go.
center
Actually-
screen now?
Yeah, that looks
Is it a
good
little
right where the
farther from that
lasers are.
sponge?
It might be. Is that the same thing or different?
They look the same to me.
The same, yeah. Go ahead, Ed.
Okay.
That's
That's
not right.
Chrysogorgia.
That's not right. I gotta do the right thing. I'm all caught up in my cow catcher in the back. Maybe bouncy.
Where you hit a cow.
Caught one.
Did we ever look at that stick? Was that here?
What's that?
The stick? Remember
I
we
think
thought
it was
we-
back a bit.
It was back? Yeah, it must have been a stalk of some sort.
Could have been a sheet flow crumpled.
Yeah. Trying to get a toe in.
You want more zoom, Emil?
Yeah.
Go ahead.
Yep.
Crinoid on it.
A little crinoid too.
Yeah.
This one looks different.
That's a
Does,
full
doesn't it?
zoom.
Oh my
so-
gosh.
Those polyps aren't
It
the
is
same,
different,
I don't
isn't it?
think.
This is
Yeah.
not a deluxe landing
It's
zone.
not as dense. Well,
doesn't
maybe
have the
it's just the-
It's
delicate,
working.
the delicate stalks of. This is different than the last one. I think. Maybe
How about
I'm
the one
wrong.
just to the right of this? Ay, ay, ay.
Let's check out the one to the right. I think those two are similar. I think what we might want to do is sample that Chrysogorgia that we saw
The one at
earlier.
the base of the sponge?
Crab or
I
a
think
hermit
so,
crab
yeah,
like
unless
that.
we've got another
Yeah.
one like it. That's closer.
I can't tell. Hold on.
Little hermit.
Is that hermit? Yeah, it is.
Sorry, Ed, that's all you get.
Yeah, I can work with this.
So what's the plan, Emil? You want to
Um,
sample the one at the base of the sponge?
Yeah, unless there's some closer to you that are similar.
Your guess is as good as
There
mine.
was a few
Your guess is-
between these and the sponge that
All right.
might be similar. Like these 3. Or 4
Can't
of
really
them.
land and look. I might be able to.
Is there one on the bottom,
What do you
maybe?
do here? Maybe I can fit this way, actually. Okay, can you zoom in there, please? Check out these two.
Yeah. Yeah, this looks like the same thing.
I'll bump down. This is or is not the one we want to sample.
I'm not 100% sure this is the same as what we saw at the base.
We can just hop back over to the base.
Yeah,
That's
let's
fine.
go back to the base.
Yeah, come wide, please.
back.
I think we've got a variety up here.
You can see the inner jaw and outer jaw print.
Yeah. This is an awful landing spot.
Sponge is pretty close on the right.
What's that?
Yeah, the sponge is pretty close on the right where the craft is.
Yeah,
Yeah,
if the
and-
sponge is too close, Steve Oskovich thinks that other one we just looked at is the same.
Oh, let's go there.
Yeah.
Less possibility for destruction.
And we'll also trigger a Niskin here.
Okay.
I think we have 4 of those left.
Yeah. Yeah, one of those 3.
The 3 amigos.
Yeah, the one,
We're going to be slurping this?
Oh, we have 1 left.
7. What do you
What's-
want to? Yeah,
we
what do
could-
you guys want to do with it?
I don't know how well this, let me check.
Are we looking to sample for associates of the Chrysogorgia as well?
Well, it couldn't hurt. We could try, that looks like it would fit in the slurp, huh? Can we zoom on the lower one?
Lower one. Yep,
Lower one.
roger.
Right.
Go ahead.
Yeah, it's right.
Yeah,
in
roger.
there.
Yeah, I just kind of screwed that up.
That lower one?
Yep.
Pardon me?
Yep. Is that some kind of associate in there?
Yeah, the pink stuff in the center.
Shrimp maybe?
Yeah, maybe.
Guess we'll find out.
All right. Yeah, what a snip and slurp.
Snip and slurp, aye. Okay, full wide, please.
So not full organism, just partial.
Partial should be okay.
Okay.
Is it small enough to fit in the? You think the whole thing would fit in the slurp?
No. Unless
Yeah.
it narrows itself. Depends how brittle it is.
Yeah.
It's gonna be hard to get regardless.
Yeah, it's quite in there.
Let me come in.
Yeah, please.
Let me come in to about there.
Perfect.
Which jar is the one that's free?
7.
I didn't hear.
7.
7 is free, Roger.
You're hung up on your lower jaw, I think.
Yeah.
Just a real gentle hug.
Do you think slurp is best at this point, or that's pretty big?
That's pretty big. I don't think slurper's best at this point, no.
Yeah, let's put it in a drawer.
Okay.
In the, which drawer?
Stand by.
Let's see.
I don't, well, I don't want that. If I was really keen, I'd pull a Niskin right now and confuse everyone.
With it in your arm.
With this in my jaw, yeah.
We put it in with the rock.
Do what?
Let's see
Yeah,
here.
we could. I'm just afraid the sponge will come out of E.
Yeah, how
but,
about a rock?
Let's see. I mean,
The sponge was sinky when we turned the thruster off.
Yeah.
There's no guarantee of it not coming out.
Okay. Let's put it in with the rock. With 74. Yeah, the
So I
sponge
could-
might float out. Yeah.
put it in starboard F.
Starboard Foxtrot. Okay. All right. Let's see.
Here, I'll let George watch this so he can. Oh, actually, maybe I can't. Let's try it. Or
Okay, can I get 4, please?
bridge video. Can you see that? Copy.
Okay, go ahead. And we're going to Foxtrot, so we'll do a halfway open.
Unless you think it'd fit in one of the smaller
Good there.
bins, but F will work.
It would fit, but then we're exposing more floaties.
Yeah, let's
So.
just do F.
What's in the port one there?
That's 080.
All right, you can close it.
All right.
Then we'll do a Niskin after this.
Yep.
And for those of you watching us at home, when we take water samples with the Niskin bottles, that's going to allow us to find environmental DNA, also known as eDNA, which could be indicative of the biodiversity in the area.
I can do 8, correct?
Yes. Yeah, that's correct.
I've got eyes on it.
Thank you.
It's very shadowy up there.
Yeah. 8 has white tape on the top of it. 7 and 8 do. There you go, triggered.
Bing.
I was going to give it a little more after in case just the top goes.
Oh,
I've seen
yeah.
that before, just the top triggers and
Not the bottom.
not the bottom.
Well, what kind of a water sample would that be? Sorry, water.
Single-sided.
Single-sided water.
It's only half as
sample.
good.
That's how they used to be. There was a bottle type before Niskin bottles that were closed on one side,
Not the other?
not the other, and then that's how they used to take water samples. And then someone was like, "Hey, we should do both sides so water can flow through it."
Oh, I understand. It was permanently closed on one side.
Yeah,
Ah.
But, and it had a name like Niskin,
It
but it
like
wasn't a Niskin.
Naskin or something.
Yeah, yeah.
it was like, what?
There you go. Okay, are we done here?
Sorry,
Sure.
can't hear you, Emil.
Did we get the Niskin? I was chatting
Yes.
with
Sure did.
Gina. Okay. Yeah, we're done. I
All
think.
done.
We're gonna be going 3, 4, 5.
3, 4, 5. Let's go.
I'll wait till you want me to do it.
Yeah.
Bridge now.
What do we have there on the left?
1 step 40 meters, bearing 3, 4, 5.
You just swiveled past some on the left there.
Pardon me, Emil?
Did you have to move on right away?
We just put a ship move in.
Yeah, but it'll take a while, right?
I'm behind, so I gotta
Oh,
go.
okay.
So yeah, do you want me to not call the ship moving, Emil?
That's okay. There were a few larger corals back there, but I think probably bamboo.
Okay.
These look nice and crusty, these rocks.
You're gonna have to come all the way around. We're going 3, 4, 5.
So, one of our viewers wants to know, do you ever find that storing or preserving our biological samples in ethanol can sometimes be detrimental to the quality of the tissue, or does it always work in terms of preserving it until we can test them?
Well, I haven't seen it on like the other end where it goes to one of the repositories, but from my understanding, I think it would preserve them pretty well.
Do you know how long they would last in that solution?
That's a good question. Yeah, I'm not too sure about that. I know it definitely would preserve it enough to take DNA
Yeah.
samples and get an ID on it. So.
Hi from the lounge.
Hello, Alexis.
So ethanol does really well for preserving for DNA. A lot of natural history museums have samples that are hundreds of years old that we can still get DNA from. For really high quality DNA, it's even better to keep them cold. That preserves the DNA in even bigger chunks. And then if you want to preserve the tissue for like morphological analysis, some specimens it's better to preserve them in formalin or formaldehyde because ethanol can degrade essentially like the morphological components, but we don't use a lot of formalin on board.
Okay. Thank you.
Formalina. That's what they call it in the Galapagos.
Has such a particular smell.
It does have, yeah, very particular.
And Alexis, if you're going to be watching for a little bit, just shout out if you see a zoom of interest.
There's a
Oh,
purple
Georgia?
one.
This is a zoom of interest for me, is it?
yeah,
of
let's
interest to
zoom it.
science?
Let's zoom
Okay.
on it.
Go ahead, Ed. All right, close enough. It's taking it out of the light pool again. I'm gonna back away.
Strong current.
Maybe that's worse.
Got darker. Got a lot
Yeah.
darker.
That's less good. Ugh. Yeah,
Okay.
not doing very good. Okay.
Something there.
Yeah.
Is that a
Let's check it out.
black coral? Can't tell from this far.
That might.
That was just going by shape, not skeleton.
Yeah.
But that doesn't, I don't know if it is. Plexaurid?
Primnoid, maybe?
Yeah, sorry, that's I think what I meant.
Okay, Ed, go ahead. Some polyp action.
I'm pretty sure it's a primnoid, but I don't know what type. We haven't sampled a
My P's
primnoid
mixed
yet.
up.
That sounds like you're thinking about it.
Yeah, yeah. I'm not getting any feedback from the shore, but yeah, if we've got time.
We can hold, sure. I think we're holding on sampling.
Okay.
Yeah, can you snip
We can snip.
upper right?
I think I'll do 20 meters back.
Sure. Pretty tall. Can I go here?
Bridge now. 1 step 20 meters, bearing 165.
Yeah, I think we could probably snip and slurp.
Yeah, snip and slurp. This is a good snip and slurp candidate.
Science, whenever you have time, I have a weird question for you.
Yeah, okay, one moment.
Yep.
I can try and get the branch that the right laser's on.
Yeah.
Do we have good
was on.
images?
Yeah, do you want more images?
Should we zoom a little, or?
Fine by me, go ahead, Ed.
Just right on that branch that you're going to cut. Yeah.
Give it a, oop.
No, that's good.
It's always good to get good photos because they don't look like this in the lab.
The before versus the after.
Yeah.
Okay, come out, please.
Nice.
I just got set up. You got that?
Yep, that's good.
Okay.
Wow, that is a good zoom.
Yeah.
Nice dark background.
Ooh.
So you said jar 7 was open?
Yep, 7 it is.
Can I come back at it? No, I can't. I gotta come this way. Can you please tip bubble up? See if you can line that branch up.
This is a Calagorgia, Gina.
Sweet. Yeah.
Gotta go this way, I think. This is awkward.
Should just, can you reposition the vehicle?
Probably should.
Now you're committed.
Yeah.
Yeah.
Yep.
And so it is a primnoid, Calagorgia, maybe Calagorgia crescentis.
All right, can I get preset 1? I'm gonna do something absurd here and just go over here. Like, that'll be bumpy as heck, but maybe it'll work. Let's try that. Okay, could you line that up again on bubble, please?
May come in.
Yes.
Right there.
Perfect. Is that okay?
Yeah. All right. Will that slurp?
Okay, come wide. I think so.
We're doing slurp.
Hard Ed, push in, please.
Yeah, stand by.
Avery's already set up.
Bucket is starboard.
Uh,
yeah.
I think so, yeah. It's too bad I gotta do it this way, though.
Can't
So
you spin?
I could, but then it's
right,
gonna be
then it's gonna get stuck.
barbed.
Yeah.
See what happens.
What kind of percent do you want?
Actually, can I get a bubble zoom on there, please? Right on the tip of the hose.
I'm starting to see a little bit of flow.
Yeah, more zoom, please. You can come like right in. Keep going. Keep going. Good there. And a little bump down. Good, yeah. And I think we might have to do 40, maybe 50. We'll start 40 on this one. So I guess let's try that, see what happens. You know what, let's start with 50 just to get us through this first little bit.
I don't see any flow in there.
There's no sediment maybe.
Yeah.
Oh, it wants to go.
Oh,
does it? Does it want to go?
I don't see anything. Oh, there you go. There's some flow in there. Pieces.
Should I re-grab it?
That's good.
You
Risky
did.
business.
I did, yeah.
Yeah, just like let it sit there. It'll. There it is.
There we go.
There we go. Cool.
Awesome.
Come full wide, please, Ed.
Full wide eye.
Let's see if it gets in there.
Come on, make it.
I have to give it the beans.
Might have to give it more beans, yeah.
Wiggle
Yeah.
waggle.
082.
Yep, 082.
Yeah, more percentage, I think, please. Huh.
Primnoid.
Yeah, Caligorgia. Just gonna give it a shake.
Yeah.
Williams.
No sign of it yet, eh?
I haven't seen
No,
it.
just little pieces from before.
Oh,
You're
now I
getting
got a lot of flow.
flow, but no pieces.
Hmm. Let's turn it off, turn it back on again, maybe. I don't know. Okay, go ahead. Maybe let's give it full beans all the way to 100.
There it
There's
is.
a little piece.
Hey.
Two
More,
pieces.
more, more. Give me more. Never really understood that. If it can fit through the first part, that's the smallest part of the whole system. Like,
There's angles it has to go through, right?
totally, but they're like big diameter.
Yeah.
Tap it on a rock.
What about
Huh.
side to side rather than, yeah. Give it the stretch.
Does not want to go in.
So nothing, eh? Or just little bits of it.
A few pieces. At least 3 sections, but certainly not the
whole
whole
thing. Huh.
Well, there's no more slurps.
So that's kind of okay.
So it's like, if it's caught in the hose,
We could
We'll just get it out
extract
later.
You try rotate to flush and see what happens.
Sure, yeah, so kill it and then spin it, I guess.
But
Yeah.
those will go through the flush, or they have a potential to go through the flush
You
holes.
think?
Yeah, I think so.
Yeah, so I don't know if we want to do too much suction on that.
No.
Maybe move it back to 7, try it again?
Sure. You're at 40% still. Yeah. Try that.
Maybe it just wasn't seated right.
Could have been, yeah.
There's flow now.
Small pieces.
What's the extraction process if it's in the hose?
Usually flushing it out works. Filling with water and dumping it, going backwards. If that doesn't work, we gotta take it apart until we find it.
Siphon.
Well, if that's the final slurp, then maybe that's what we do.
Okay, we can keep it here for a bit and keep it running.
Okay. Can we look around?
Sure.
Make a note of that, Gina. Might have to search for it in the hose.
Yep. Putting it on there.
Okay, we ready to look around in this area?
Yeah.
Okay.
Okay, it's our collective job to not forget that the slurp is on.
Yeah. Collective.
Science, do you know if we're sending any of our water samples to laboratories doing metaproteomics? And that's a new term for me.
Metaproteomics.
Our samples just go to a repository.
It's a new term for me too.
The water, though, gets processed for eDNA. It goes to Meredith Everett's lab.
Where?
EMEL. Am I mistaken about that?
That's where it goes.
I think that's
Yeah.
correct, yeah.
The filter goes to her lab.
Yes.
Is there urchin down in there?
Yeah,
Yeah.
nice purple urchin. Yeah.
What if maybe the slurp hose clear?
Yeah, metaproteomics, according to the interwebs, is the large-scale identification and quantification of proteins from microbial communities.
Yeah, to my knowledge, we're not using the water samples for that.
You okay with starting moving again, Emil?
Yeah, I am. Yeah.
Okay.
So you've got you're stepping directly towards waypoint 3, Rennie?
Negative. Just going up to here, to this flat bit.
Okay.
Do you want me to
Maybe once we get to that contour that is the same as waypoint 3, we could follow that along and then go up there.
go up? Okay.
Yeah, and then go across to waypoint 4.
I'll still go 3, 4, 5 there
Yeah.
till we get there.
That's good.
That cage just moved a lot.
Yeah. The black one?
Oh, is that your thruster? Your,
Yeah.
yeah,
Main hydraulic pressure.
yeah.
You want to see it really drop? Holy smokes, here you go.
That's not too bad.
A third of our pressure gone.
Yeah.
Can I get front porch, please?
Do we want to leave it on jar 7 or go to flush in case it's kind of 6 to 1, half a dozen the other, isn't it?
Kinda, yeah. We just top the flow.
Yeah.
So hello to all of those of you who just joined us during the last few minutes of the dive. We are currently making our way up a slope located to the northwest side of Howland Island. We are looking to get observations and samples of high-density communities, focusing specifically on sponges and corals as well as on their associates. If we see any fish swimming around, we might be zooming in on their heads to get some identification information for scientists ashore. We might also be sampling from some geological features. So we're specifically looking for iron and manganese encrusted nodules.
Let's zoom there. That
Right
guy,
here?
That guy to the right.
Right to the right.
The bonsai.
Yeah, we wouldn't expect to see any nodules at this depth now. We're just looking at encrusted side of the seamount.
All
The
right.
nodules we were looking for when we were at 3,000 meters, but
All right,
we're
go ahead.
unsuccessful.
Are we still at Howland? Can we still see
We're-
that?
I'm sorry?
Can we still see Howland?
No, we went far away from it.
Yeah, it's 150 kilometers.
Oh, okay. Does look like a bonsai tree.
Yeah.
It does.
All right, and was there interest in the one up to the right here?
Sure, yeah. Oh yeah, you were to Gorgia.
Old hat by this point.
Yeah. Never get tired of looking at it though.
That's true.
It's got an associate of some kind.
You can push in a bit more. It's got a purple one down
Two
low.
of them. Yeah.
Snail?
Snail.
What's that? Is that a tunicate on it?
The purple,
Yeah.
just hanging on by a thread.
Good, barely,
Wrong
yeah.
way.
It
Dandelion.
looks like it has two of them.
Don't think I've seen a snail yet.
That's cool, yeah. All right, come wide, please.
Hey.
Yeah, there are several of those.
Nice candelabra.
Wow.
Or rake.
Wow, this is a nice area.
Yeah.
Whoa. Look at that baby.
That's cool. Very three-dimensional.
It's like elk horns.
Yeah.
Can you swivel around and take a look at that?
Sure. Yeah.
So bamboo is the candelabra. You can see the joints. And is this also a bamboo?
We could zoom in if you want.
Yeah.
Go ahead.
Partial.
I don't see any bamboo joints.
I'm not seeing the joints.
Yeah.
Coming in.
Bridge now. Add one step.
And focusing to the back.
I'm not sure what we got here.
To the one near us.
Cool.
And to the back.
Nice.
We get the whole thing in view again?
Yeah.
So maybe there are some faint joints on this, Meredith is saying. Maybe a bamboo. Yeah, I can't think of anything else it would be.
All right, should we move along?
Let me see. Do you have a ship move on?
I
Yeah.
do.
Yeah, we'll move along.
Roger.
We're going 3, 4, 5.
Thank you.
Bridge now. One more step.
Could you come down on Delta, please?
So this might be too much of a generalization, but one of our viewers is wondering if it's unusual to not see any significant life, or I guess density of life at these depths.
Unknown.
Yeah, and in this case, we're exploring an area that has never really been visited before. So we have an idea as to maybe how the currents might be influencing the organisms that live there. At these depths, we don't have any photosynthesizing organisms. We don't have any access to light. So the animals that are growing along at the bottom are solely reliant on all those currents and the influx of nutrients and detritus, all that decomposed organic matter that's coming up to be able to feed them. So we're trying to focus on observing and trying to find those little hotspots of life, but there's really no other way to tell, I guess, whether we're going to find it or not.
We tend to look along ridges and topographic highs, you know, that the top of a mound where currents tend to accelerate there. Tidal currents sweeping past will accelerate over those features, and that's usually favorable. So we're ascending a ridge at this time for most of this dive, and so with that in mind, and so But yeah, the corals and sponge communities tend to be patchy, and it's still determining why they're very successful where they are. It's got to be a combination of the currents, the substrate, and the nutrients available.
But at this depth and temperature, it's not going to look like a tropical fish tank down here. You're not going to have schools and schools of fish by any stretch. We are 1813.
Yeah.
Oh, it's a rock.
So 1,812 meters at this time. So just
So
keep
it's-
in mind that all of these organisms need to be adapted to live in really cold environments very often. And there's not a lot of food available often, and that would be mostly for the fish. And
Yeah.
they have to withstand really high amounts of pressure.
Yeah, pretty certain that that was a bamboo that we were looking at, one of these internodal bamboos.
Yeah, we are 1.125 miles deep right now.
That's really significant.
Yeah, weird.
Is it 1 atmosphere of pressure every 30?
Every 10 meters,
10
yeah.
meters?
Plus one at the surface. We're 1813.
181 atmospheres.
182 maybe, yeah. 14.7 psi for each. That comes out to, hold on, let me think, carry the 3 plus 7,
One.
more than 1.
Greater than 1.
That's a good idea.
If there's anyone joining us for the first time during this cruise or season, you can go to the expedition tab on our website, and there's a really great overview of all of our cruises and the season so far. So you're going to have an overview of the focus during the cruise. And there's also highlight videos and pictures available on our website from all of the exploration that we've done so far. Is that white structure on the bottom right like a coral base?
A piece of it's a skeleton. Yeah, looks like a skeleton of bamboo.
Oh yeah, the little piece that's detached on
Yeah.
the bottom.
What's this on the left? Kind of a scrub brushy. Oh, it's probably a crinoid.
Crinoid?
Stalked.
Oh, is it? I think it's a coral.
Looks pretty big.
And a crinoid.
Oh, crinoid behind a coral.
2 for 1.
It looks more spaced out polyps. Am I seeing that right? Can you zoom in,
Yeah.
please, Ed? Cool.
All right. So is that navigation data accurate? It looks like Herc is in front of the ship, right? Leading the way? Or are we catching up?
We're catching up. We're behind.
Okay.
We're going north right now.
Yeah, and then so is Argus behind the ship?
Both Herc and Argus are behind the ship.
Okay, so they got to swing, huh?
Yep. I'm holding here for a bit while we get a little bit more, and then I'll go this way.
Great. Yeah, it's safe because we're going along the contour.
Yeah.
The current came down eventually.
That's good.
Yeah, that was good.
Does anyone know if there's any species of whales that will come down this far?
Beaked whales,
Beaked whales could come down this far.
possibly sperm whales.
So when they do so, are they just looking for food sources?
They refuse to give interviews.
We think
Yeah, I
that
don't know
maybe
if-
that's
yeah.
why they gouge the seafloor is maybe looking for food. I don't know.
Or it could be to get rid of parasites on them.
Yeah, I have no
Nobody
knowledge
knows.
of their behavior, so. So if there's like any ectoparasites growing on top of their
I don't know.
tissue, maybe
It's
they're
only on
like
the
just
bottom.
scratching
They're very
it.
small. Yeah, you think they do their whole body or something. But there's lots of interesting ways to sea creatures get rid of parasites. Cleaning stations.
I love
Mola
cleaning
Mola
stations.
use seabirds to do it. Which are actually, what are Mola Mola? Pacific sunfish?
Yeah.
Yeah.
Why are there two different names? Just common name and real name?
I'm going to guess that one of them's a Polynesian name just because it repeats itself. And that's not uncommon. I don't know.
Cool.
You know, like poo-poos?
Or humu humu nuku nuku apua'a.
The state fish of Hawaii.
Trying to see if I can find the definition of mola.
That's smoothing out
There's
a
a
bit
lot
now.
of fish
Yeah.
that would sound cool if they were named twice. Flounder, flounder.
It says here that it's the Latin word for millstone.
All right, we'll try it. We'll try 015.
Huh.
Yeah, I don't think it's derived
It's
from the
gonna be a
Latin.
bit.
No.
What do you
'Cause
think?
we're going to go to 015 to here, but we'll try to have Herc there and Argus behind.
Sure,
But
yeah.
if there's any messiness, the
Messy mess.
messy mess.
Messiness, yeah. Sure.
Bridge
Messiness?
nav,
What kind of mess? The
one
messy
step
mess.
40 meters, bearing 015. Thank you.
Are you able to pan left briefly? Okay, it's all right. I saw that skeleton, that single stalk there. Thought it. Oh, there's a nice big Iridogorgia.
Coming up on the top of the ridge here now.
Is this all hard substrate here, Rennie? Can you check the back scatter real quick? I'm thinking maybe we could slide left and then go north.
Should all be hard. It's just, you know.
You got a 015 dialed in right now?
Yeah, going to waypoint 3.
Yeah, I'm thinking maybe we just follow that the ridge rather than the side of it, because we had a pretty high density there on the beginning of the ridge.
Okay. Yeah, this backscatter doesn't really look so.
Yeah.
Doesn't look so hot.
Why is the ridge just less?
Well, I don't know. It's quite linear here, and I'm wondering if that's due to this is the direction that we.
Oh yeah, okay.
So, yeah, so I'm not trusting the artifacts in there, and also the slope and whatnot. So.
Right.
I don't think at this scale it's helping us. This scale at this depth, but could be wrong.
Yeah, so the next step, let's go directly to that topographic high and then cut over to 4.
Okay.
Oh.
Cool.
More branching nodal bamboo, I think.
Yeah.
And turn up, but that yellow is strange.
Let's zoom in on the yellow, please, Ed. Fantastic.
That's bamboo,
color.
yeah.
Go ahead. You were gonna guess 352. Yeah, I was gonna go 345 because I'm anticipating where Argus will be once we complete this move.
Look at the dead bits in there.
Oh, that was your guess. I see. No, you're not far off. I think I'll do 345 though. 350. How about that? Compromise. Well, it's all a guess because Argus isn't settled, so it's kind
What's
of
that
moving.
there?
What are you looking at?
So we'll go
Basket star?
350.
Yeah, I
Urchin?
think it's a basket.
Basket star,
Yeah,
I
thank you.
love those.
We take a look.
Yeah, we'll just have a really strong pole
Good,
attached
yeah,
to Argus. That's a good idea.
Zoom in on the
George.
star thing.
Yeah.
And you can put that move in, thank you.
No, I
Huh.
don't know.
But
Don't know.
Almost
Is that
looks
a
like
Brisingid?
some version of a crown of thorns.
Yeah.
Or no, it's just a sea star, isn't it?
But it's got a big
What is that,
bulbous
a
bit.
sea star on something? Or is that 2 organisms? Is that some eggs and a sea star?
I don't know. It
Asking
almost
a lot of
looks
questions
like
over
a
there, Ed.
sea urchin's test.
Little
Right.
It
bottom?
does.
Nah, not really.
No?
I'm not buying sea